Continue. Okay, and let's start. Let's start the second day on of the workshop, and that's the QCB W26 workshop on protein structure with AlphaFold, and we'll continue on. Let's see. Yesterday, we went over some protein structures just to look, just to see how proteins look like and where to find those structures, how to display them on a screen in a version different than columns with numbers. And we also went through some common features to protein structures. We'll continue on with a couple words on how do we learn about protein structures, and then what determines the way they look. And then we'll squeeze a little bit of protein protein structure prediction, the simplest case, the homology modeling. We'll try to see how Swiss model website works. Okay, so as far as the recap goes, we covered the very basics of protein structure. This should be for most, if not all of you, just recap from some intro to biochemistry. That's pretty standard stuff that probably every one of you knows by now. But just to remind everybody, proteins are polymers of amino acids. They are linked by atoms that form a backbone. There are repeating peptide bonds. Here there are planar, and they connect individual amino acids that are going along the sequence. The backbone structure is common to all the proteins. There are particular side chains. The sequence of side chains differs between different proteins, and it gives them different properties. When you look at what type of side chains you can see either hydrophobic side chains. That's the most of them. They are composed of carbon atoms with hydrogens in various arrangements and then various numbers. And then in a couple of cases you've got polar groups, but overall chain is dominated by a large apolar part. That's in case of tyrosine and tryptophan. You've got those two or one aromatic ring present there. There are also side chains that are charged. There are 3 positively charged, two negatively charged side chains, and then few of side chains are polar but not charged. That's serine, threonine, and two amides that are related to the carboxylic acids, aspartic acid and glutamic acids. And then there are 3 special cases. There's a cysteine put aside because of unique chemistry of SH group. Among others, the SH groups can form disulfide bonds. They can make connections between side chains within the same or adjacent protein chains. And then there is a amino acid that's missing a side chain. It's got just a hydrogen in place of anything bigger. And then there's a protein where side chain actually bends back on the backbone, so it forms a ring. That's what determines so-called primary structure of the amino acid sequence, the order of the side chains. And then if you start to look into local arrangements of how the backbone folds locally, then you're gonna see that there are repeating patterns. There are common to most common are alpha helices, which are periodic with they form like a helix. The backbone makes turns and then twists around itself. The structure is, as I mentioned, stabilized by the hydrogen bonds formed between the carbonyl group of a main chain and the the hydrogen NH group from a few residues in front of it, behind it, depending which direction you're coming from. The other arrangement of that's common are beta sheets here. Individual strands are straight. Don't they are not periodic. They don't fold back on themselves, but they are more or less straight, and they most often occur in more than one instance. You're getting, in this particular case, 3 of those, and the entire series of those so-called beta sheet is stabilized by hydrogen bonds, now connecting to adjacent strands within beta sheets. In this particular case, we're having strands that are parallel. They all point in the same direction. Those are all C-terminal ends of the strands. They can alternate, and that's another variant of beta sheet, but the idea is the same. You've got straight stretches of backbone connected together by hydrogen bonds in between. Now, once we look from further distance, we're gonna see that those secondary structural elements can be arranged in repeated ways. That's how entire protein domains or folds are described. Initially, we look at how secondary structural elements are arranged in three-dimensional space. This specifies so-called class. And first, we look at which types of secondary structural elements show up. You do have group of proteins composed mostly of alpha helices. There you've got a group of proteins composed mostly of beta sheets, and then there's architectures where those things occur together. Now, for each arrangement of secondary structural elements, we can look at the different ways those elements are connected. For each of those classes, we've got different arrangements of the secondary structural elements in space. That's describing the architecture. And for each of specific arrangement of secondary structural elements, like here we've got a couple of alpha helices on the right side, couple of alpha helices on the left side, and beta sheets in between. We can connect them in different way by the loops, and that's corresponding to different topologies. So that's how protein domains are classified. We're starting from classifying them as belonging to one of the classes, depending on which secondary structural elements occur. Then architecture specifies how they are arranged in space, and then finding topology specifies how those elements are connected together. This covers just one domain, one protein chain. When we look yet and step far away, you'll see that protein chains can form complexes. This is so-called a quaternary structure. We can have different arrangements of individual protein molecules. Here we've got the primer of identical chains. Here, in case of hemoglobin, we've got a tetramer with two chains being of one type and two chains one type, two chains of the other type. And here there are two more complicated cases here with the two different protein chains forming a 8 to 8 complex, eight chains of one type and 8 chains of the other. And here's even more complicated, less regular pattern where we have many different chains forming together big complex. Apart from combining combining domains by forming complexes between distinct chains, the domains can be forming along a longer piece of chain. You can imagine that in this particular case, you can imagine that the sequence of the purple chain would be fused to the sequence of this guy, and then in turn to this one, you would have a series of three identical domains connected by the loops. That's another common way of combining domains into bigger arrangements, and it could be that those repeated subunits are nearly identical. They correspond to multiple copies of a very similar folds, but you can have a entire collection of different domains along the chain, and then you're gonna have different, for example, different functionalities, different enzymes combined into one within one longer chain. So that's another way of combining uh, individual structural subunits into bigger entities. Okay, so that's in a very brief way description how proteins look like. Now, how do we know that they look like this? I mean, in our case, we went to PDB and got records with information about individual proteins, but where they came from. Well, originally the oldest method there that was used to determine protein structures was X-ray crystallography. This goes back to probably initially like 1930s or so. It took about 20 or so years to actually get to the first protein structure was determined in the mid 50s. And the idea is relatively simple. You just take protein, you purify it, and then you increase its concentration to extent where it starts to fall out of solution and form protein crystals. And with a little bit of luck, you can end up with crystals that look like this. They can have different shapes, different forms. They are hard to get, and this is the limiting factor in terms of that slows down the structure determination by X-ray crystallography. You've got to spend quite a lot of time just trying different conditions. There's no hard rules how to make a given protein crystallize. So you very often resort to trial and error. If you're lucky, you're gonna get nicely looking crystals. Then you isolate one of them, and then you put it in X-ray beam. And once more, if you're lucky, if the crystal is of high enough quality, then once you shine the X-ray beam on a crystal, you're gonna see diffraction spots. The reason is the crystal is a regular arrangement of protein molecules in a three-dimensional space, and it acts as a diffraction grating. It diffracts the light. In our case, we're using X-ray beams, which are electromagnetic waves with a very short wavelength, much shorter than the visible light, but matching the typical distances within the crystal between individual atoms. And if that's the case, then the regular arrangement of atoms acts as a diffraction grating and results in diffraction image, the spots that you observe on a detector. This is just a diagram. If you want to see a real data, that's how it looks like. You're gonna see a collection of spots. This is for one orientation of a crystal. This is where the center of the beam is. This is where it hits the screen, and then farther away from the center, you're gonna see spots of various sorts. When you rotate the crystal a little bit, the pattern changes. This is, I think, half a degree or so rotation, and you see the pattern changes, and then another half a degree, and there's another image. If you look into some reflections here, then you're gonna notice that some of them are stronger, some of them are weaker, some of them are yet weaker, and some of them might be not there. The spacing between the spots, there's repeated distance between the spots along this line, it's same as here. And then you've got spacing between those lines. There's also spacing in the third dimension. The spacing between the spots tells you what's the arrangement, how, in what way the proteins are repeating within the crystal. It tells you what's the so-called lattice of the crystal. Whereas the intensities that you see here, the different intensities, tell you what's the shape of what's inside of the repeated unit. So to get the idea what's the shape of our protein, we've got to record those intensities. It's not as straightforward as you might think, because each spot's got a slightly different shape. There's a different background that you see here. It's not like the entire area is of the same intensity. It's much brighter here, much darker here. So you've got to somehow take into account the background behind that's contributing to intensity of this spot. So it's not quite straightforward, but it's possible. At this time it's done by program, so you don't have to do it by hand. And at the very end you end up with an identifier of a given spot. It's provided as a set of indices. Three numbers specify which of the spots you're looking at. And then for each spot, we do have intensity, how strong the spot is, plus we can estimate in some way the error, how accurately you can measure given intensity. Obviously, this one is going to be measured much more accurately. It's much stronger relative to the background, versus intensity of this reflection is much weaker. So the error is going to be much higher. So that's what we can extract from the experimental data. The problem is it's not all that's there. There's additional parameter that's characteristic of this image, which is a so-called phase. If we look, you've got to remember that this spot is generated by electromagnetic ray hitting hitting the screen in this position, and this is a this is a this is a change in a electric or magnetic field over time. So it the intensity of this spot corresponds to how strong the oscillation is, but it doesn't tell you about timing. This is a very high frequency. We can't follow this oscillation by measuring the intensity of the field along the time. We're just measuring the amplitude. And for this spot, it could be the oscillations are happening in such a way that at a given time we're somewhere here along the sine wave, but here it might be the same, but it could be different. If we shift the wave a little bit, then amplitude is going to be same, but we can vary the timing, the phase, so-called phase of the ray hitting the spot here might be different, and we can't tell those two things, two versions apart. What it means is that we're getting only half of the information. So most of the effort in the early days of X-ray crystallography was spent on figuring out ways of actually figuring out what the phase of those individual spots is, because this is required to get get our structure. Assuming we know both our intensity and the phase, we would be able to get the structure. There's relatively straightforward math that gets you from a collection of intensities and phases to the structure. Problems, we don't have them, so we've got to somehow figure out what to use. Luckily, it turns out that if we've got the fraction image and phases, assuming we've got them, we can get to the structure. But it's also true that if we do have a structure, we can go back and retrieve both intensities and phases. If we know the structure, the reverse path is there. So the simplest case is to get is to use a structure of a similar protein and use it as an initial model, use the phases from this model and experimental intensities, and then we can calculate where our protein in a structure we're trying to solve it actually is. And that's the case here. This is actual reconstruction of electron density using a model here in purple, which is known beforehand, so we can place it in our unit, our crystal, and use the positions of the atoms that are coming from the model to calculate the phases for the reflections. And then when using intensities from our experiment, we're going to see some additional places in green where the difference between the initial model and our structure is. We can then use those differences to place improved model of our protein there, and then once we've got this better model, we can recalculate the phases and get the better phases from the model, and then those combined with the experimental data will get you actually the structure of the new protein that we're looking at. This type of approach is called molecular replacement, and this is one of the reasons that PDB is quite popular within among crystallographers. Every crystallographer, when you're trying to determine a structure, when you've got the crystal, the first thing to try is to see, do you have a model already there of a protein that is similar to ours, and then we can use that model to solve so-called phase problem and get our structure in a relatively straightforward way. If there's no modulus, there are other methods which are more complicated and more harder to explain. But if you feel like it, you can take a class every winter. Structural biologists here in Bayer hold run a class. It's a M213B and M213C and D. This is a combination of lecture and a class, and they explain all the nuts and bolts of X-ray crystallography and also of cryo-EM, which is another method used to determine protein structures. So this is a very brief information about X-ray crystallography. Once more, this is entire separate story and series of lectures. Typically, it's taught over a quarter or so. So there's no way to explain everything, but the idea is essentially, as I said, you purify your protein, you grow crystal, you shoot X-ray beams at your crystal, you collect the diffraction data, and then convert it into a structure of the individual molecules in your crystal. Now, the method that's becoming nowadays very popular. That's last 5, 10 years is cryo-EM, or her full name is cryogenic electron microscopy, is a method which starts with preparing your sample as before. You've got to somehow get your protein complex in a test tube. But in the difference between X-ray crystallography and cryo is that here you don't have to grow crystals. All you do is you place a little drop of your protein solution on electron microscope grid. This is essentially a metal support with openings that you're gonna shoot the electron beam through to look at your proteins. You put a drop of it on a EM grid. You blot most of the solution away, and then you rapidly freeze it, typically by plunging the grid into ethane, liquid ethane at liquid nitrogen at -80 or so temperature. Is typically liquid nitrogen, and or even lower, and then the water in your sample rapidly freezes without forming crystal, and then traps the individual molecules of interest within the frozen so-called vitreous ice. And if you squint, you're gonna see a cross section of a EM grid with your sample. There's a layer of water in the spacings between the metal support or carbon film that's used to provide a finer support with small openings. And then each window there are going to be individual molecules or protein complexes suspended, each in different orientation, hopefully. If you then put it into electron microscope, you essentially perform the same observation as with light microscope, except that you're using electrons as instead of electromagnetic waves. The advantage here is that the interaction between electron beam and protein molecule is much stronger, so you can look at individual molecules. With X-rays, you had to have an entire collection of molecules in form of a crystal to be able to record the signal with sensible accuracy. With cryo-EM, you can see individual molecules. And the way it's done it once more, you've got the source of the electron somewhere at the very top of the column. You've got the bunch of lenses that form the beam before it passes the grid, and then you've got couple of more lenses that act the same way as objective and ocular lenses in a microscope to project the image of your sample on a grid. And that's what you do. Collect a lot of images with your molecules. Each image might look like each image of a grid might look like this. There's a lot of tiny things there. If you squint and look into three of regions here, you're gonna see a very weak shape corresponding to individual molecules sitting in your sample, hopefully oriented in different way. Each one might be different in different orientation. The initial signal to noise, the initial intensity of the signal is pretty low. There's a very low contrast between individual particle and the background, but you can use a computer to actually collect images that look similar and stack them and average them together, and then you end up with so-called 2D classes, and that's what's shown here. You see different images, different 2D classes. Each one is corresponding to a stack of images that are similar added up together, the signal from the structure, it's adding up much faster than the noise. So if you have enough of those images, you can end up with a better better quality image. Those 2D classes each corresponds to a view of your complex from a different side. You can imagine that you're taking a picture of a person when walking around, and then taking pictures from different directions, and then in principle you can take those images and put them together to build a model that, if you rotate it in the correct orientation, will give you which will best return you the photographs you took. This is the same case as here. Here you do have images of your protein taken from different orientations. So then the program is taking all of those and is trying to build a three-dimensional model that, if you take pictures from different orientations, will regenerate all those images that were collected, and that's what's the initial result of cryo-EM is a three-dimensional model which will best regenerate the images that were collected experimentally. And that's what you do. You reconstruct three-dimensional shape of the protein complex from a collection of images taken by in electron microscope. And once you've got this initial blob of density shape of the molecule, then you build into this density model, and this is what's coming out of cryo. Ultimately, you're gonna have a model like you've seen in Chimera, that was built into a density observed by cryo, or determined by cryo-EM. The difference between X-ray crystallography and cryo is that with X-ray crystallography, you can go back and forth between the model and the data, and the model. So you can check in a very, very strict way if the model is consistent with the data. With cryo, it's less. It's not at the moment possible to reconstruct the image back from the model, so there's not much feedback. So refinement of the cryo models, it's much harder, and might be of less quality than electron diffraction. But there's been tremendous progress over last 5 or so years. So and at this moment the cryostructures are quality-wise comparable to what you can get from electron diffraction. So that's the second. The advantage of cryo is that you can determine structures of proteins that are very hard or impossible to crystallize. So that's a serious advantage. It's bypassing the major bottleneck of electric crystallography, which is growing crystals. Now, those are the two methods that rely on measuring the positions of the atoms in space. In both of the cases, you're going after XYZ coordinate of a given atom. The third structural method, the NMR, is not going initially after positions, but is going after distances. It measures how far two atoms are. That's essentially what NMR does on a large scale. You can record NMR spectrum, and from the spectrum you can determine what are distances between pairs of atoms. This is in principle enough to reconstruct entire structure. And here's a very simple demonstration. Let's imagine you've got 1, 2, 3 atoms, and you know the distances between each pair. So then you can build a triangle. The orientation of this triangle is up for grabs, but you can start from some arbitrary position. You don't care about the orientation of your molecule. You care what's the shape, and that's all you need. You've got to know what's the relationship, how far each pair of atoms is. Okay, and then if you've got another atom, let's say fourth one, you can measure distances between this new guy and the three that you had before. Well, this is ambiguous. You can put this atom here, but you can also put it here as long as the distances are the same. Those are those are two equivalent positions. So you can't tell which one is correct. Well, you guess one of them, okay, and then you keep going. Let's say we put it here, and then we've got fifth atom. Now we've got four reference points. Those 1, 2, 3, 4. You can have four distances, and now ambiguity goes away. Now this is the only place where we can put the atom, right? If we had only three distances, the story with flipping back and forth would hold on. But if we collect at least four distances for next atom, then we're fine. So from now on, we do have a spatial arrangement of those atoms that we can determine by just adding more atoms with more and more measured distances. And then at the very end, once we build the entire model, each atom corresponding to one particular atom within the backbone or side chain of a protein, we can then check how the structure looks like. Are the amino acids you see there the L version or D version of the amino acids, and if they are wrong, if we flipped, if we've got the wrong handedness, we just create a mirror image of our model, and this is gonna be whatever our structure is. So this initial ambiguity is at the very end resolved by looking at handedness, the chirality of the individual amino acids, and this allows us to compensate the initial guess we had to make. And that's essentially what the NMR does. The technicalities are complex because measurement of all those distances is complex. It's typically done by recording various types of 2D or 3D NMR spectra. Each peak corresponds to one particular atom. Within here is a small molecule, so you've got a relatively small number of peaks. It's still large, but not that large. And then we do see a conventional one-dimensional NMR spectrum along diagonal, but then each cross peak connects one atom with some other atom, and intensity of this peak tells us what's the distance between the atom that corresponds to this peak versus from the atom that corresponds to this peak. So you've got two atoms, and then the distance between those two, actually two nuclei, not atoms. We're looking at the nuclei of the atoms. The intensity is corresponding, depends on the distance in a pretty steep way. So in practice, we see only atoms that are not that far away. As you can see, for even a small organic molecule, the spectra are getting complicated. For proteins, they are even more complex. There are not as in the case here 30 something atoms. There are thousands, if not tens of thousands of atoms. So there are limitations on how large proteins you can work with NMR. But the idea is just like here. You measure a collection of distances between nuclei of different atoms, and then you use those distances to build a three-dimensional model of the protein structure, and that's what's coming out of NMR. This advantage here is you don't have to grow crystals, you don't have to prepare grids. The disadvantage is that proteins can't be too large. There are there are a number of reasons. One is, as I said, the large proteins do have a very complicated spectrum, so you can't sort out which peaks corresponds to which residues, and it also matters because the large proteins tumble more slowly in solution, and when proteins don't tumble fast enough, then all those peaks are becoming very broad, and you just can't see them. So there are limitations on the size imposed by the physics behind NMR. But nonetheless, there's quite a few structures that mined by NMR that 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 wouldn't be possible to obtain in other ways. So that's yet third major way of getting the protein structure. So and that's the source of the data that you see in PDB. It's either cryo-EM or NMR or crystallography. Each record will have a information how the data was collected, what was the method, what are the conditions. So you can check all of that in a PDB records. So that's where the structures are coming from. We'll make a 5 minutes break now, and then we go over why protein. So far we know how protein looks like, and we know how we've got at least some idea how we determine the protein structure. Now the next question is why do they look like this? So that's gonna be after the break. So let's make a break for 5 minutes. I'll stop recording. No, I don't want to stop. I want to break, not stop. Oh, come on. Okay, let's stop. Okay, let's resume recording and continue with our workshop. So now we're gonna go over protein folding. The issue is what determines the shape of the folded protein molecule. Why does it take one shape or another? And initially, somewhere at the dawn of protein biochemistry, it was not certain. And finally, I think somewhere in the 60s, Anfinsen actually ran a series of experiments where he was able to start with the purified active ribonuclease. This is one of the molecules that you've seen yesterday as examples of the protein structures. He was able to denature it to unfold the protein. This is typically done by adding either urea or guanidine chloride. Those molecules bind the water in a solution, and then protein unfolds. But then once you remove the nitrate, once you remove the urea by, let's say, dialysis, then protein will spontaneously go back to the structure it came from, and you can monitor by just looking at activity of the ribonuclease, and that's what Anifson did. This was the basis of his hypothesis or dogma, whatever you call it, that states that the three-dimensional structure of a protein is determined by its sequence. That's the short version. The long version is the longer version of that you see on the screen. The essence of the observation is that the protein in its native state is at a minimum of its free energy. So that's that essentially gives you a prescription how to calculate the structure of a protein. First, write a formula that is gonna tell you what's the energy of an arbitrary protein chain folded in or bent in a or placed in a given conformation, and then then keep searching for uh, for the shape, for the conformation that corresponds to the minimum, and this would be your calculated structure, and according to the Alphinson's idea, this would be the actual structure that you would get if you determine the structure experimentally. That sounds simple, except that there's a lot of caveats there. First, you've got to calculate the energy. You've got to know for arbitrary conformation of a arbitrary collection of all the torsion angles, what's the energy of the chain. To do that, you've got to go over individual contributions of the individual effects of that contribute to the energy, and those we did get to one of them, to hydrogen bonds. I mentioned quite a few times that secondary structural elements that not only are stabilized by hydrogen bonds, those are bonds that are analogous to what water molecules do when interacting with each other. As a reminder, we do have a OH bond, that actually two of those connecting hydrogen atoms, and with the central oxygen atoms. Those bonds are polarized. There's a partial positive charge on hydrogen and negative charge on oxygen, plus there are two lone electron pairs with no corresponding proton here, which are quite negative. So on one side of the water molecule, there's a negative charge. There are two hydrogen atoms that are positively charged, and those two ends of the molecules attract each other, and that's what the hydrogen bonds are. In case of water, they are formed between oxygen on one water, hydrogen belongs to it, and oxygen on neighboring water. But you can imagine, in case of proteins, that you do have a oxygen atom on one in one place in a chain. Then you've got the hydrogen down here that belongs to the nitrogen hidden behind, and the nitrogen. So you've got here three atoms: nitrogen, hydrogen in between, and oxygen the other end, and that's the hydrogen bond between the carbonyl group of the main chain and amide nitrogen at some residue, few positions apart. Here's another better visible place where hydrogen bond is. You've got the hydrogen atom is not drawn here, but you've got to imagine that somewhere here there's a hydrogen atom sitting between the nitrogen and oxygen atom down here. So those are hydrogen bonds. Those are interactions that are essentially electrostatic in nature. There's a partial, as you've seen here, there's a partial positive charge on the hydrogen, partial negative charge on the oxygen, and those two charges attract and form are responsible for formation of hydrogen bonds. We can obviously have interactions between charged atoms. Some atoms are charged not by virtue of polarizing the bond, but there's actual charge corresponding to a charge group. That's what we do have here. There's a amine group. It's gonna have three hydrogen attached that you don't see, but overall charge of this group is gonna be plus. It's gonna be +1, and then you've got the carbonyl group of some adjacent side chain that does have a negative charge. So this is attraction between a negative charge and positive charge here. In this case, also accompanied by a couple of hydrogen bonds. Those atoms got close enough so that the there's significant effect of interaction mediated by the hydrogen attached to amine group that forms a hydrogen bond with a oxygen level carboxyl group. So sometimes you've got those two effects contributing to contributing at the same time. When you so this is the case of attractive force where you've got the electrostatic interactions that pull things together. You can imagine that if you've got charges of the same sign, then those residues would be pushed away. So there are both interactions that force things, brings things together, and there are also interactions that push parts of the molecule apart, and then there's some balance in a structure that corresponds to local minimum of the free energy that determines where things will end up. Apart from those two types of bonds that are electrostatic in nature, we do have another significant contribution from hydrophobic interactions or hydrophobic effect. They are most easily noticeable somewhere in the center of the molecule. If we zoom into center of our tin barrel here and see what's there, we will notice that most of the side chains here are not polar. Those are all either aromatic rings. You've got one aromatic ring here. This is phenylalanine. You've got the tryptophan here with two condensed rings. Another highly hydrophobic residue. You've got here, I think, isoleucine or isoleucine. Yeah, isoleucine here, I think. Here's another one. So most of the atoms within the protein structure are not polar, and this is due to so-called hydrophobic effect. How does it work? Well, if you take an apolar molecule or atom, solid in general, then that cannot form hydrogen bonds, the water around it will orient in such a way to form not so that it doesn't waste the chance to form hydrogen bonds. So this molecule here will orient itself in such a way that it will be able to form hydrogen bonds only along the surface of the molecule, the apolar molecule, to make connection to other water molecules. So in effect, the apolar solids are surrounded by cages of partially oriented water molecules. This thing is decreasing the entropy of the system, and that's not favorable. In general, systems try to increase their entropy according to second law of thermo. So what happens is when you do have, let's say, two apolar entities, two solids in a solution, each surrounded by a shell of immobilized water, then you'll be able to make water less ordered. You'll be able to increase the entropy of water by merging them together into one bigger blob, and then whatever water was immobilized on the surfaces that came in contact will be free to move, and they will contribute to increase of entropy. So the hydrophobic effect is essentially driven by the solvent. It's not a property of individual molecules that are hydrophobic. The only feature they've got is they cannot form hydrogen bonds with water, and then the rest is because of what's happening with water. It's properties of water that actually drive those greasy things together. This makes computation of the energy of the protein chains much harder, because it's not enough to characterize the protein molecule, all the side chains and whatever properties you've got there. You also do have to model the properties of water. And as I mentioned yesterday early on, if you look at the protein structures determined by X-ray crystallography, you will see all those water molecules present in the crystals. They are in principle parts of the molecule. They contribute to the energy of the protein chain. So it's very hard to model the additional contributions of all those water molecules, and that's one of the reasons the accuracy of the energy calculated for a given protein conformation is pretty hard to get. It is a limitation on a way imposed by the way we model the protein in a solution. So this is one limitation, but oh, and just to complete, there's a the fourth term that's physics based, which is so-called van der Waals interactions. Those are interactions between the electron clouds surrounding each nuclei. If you push two atoms too close together, they will start to repulse. They won't let you to pass one through the other due to electrostatic interactions between electrons in electron clouds, and that's the basic of the van der Waals interactions. They are typically modeled as so-called Leonard-Jones potential. This is the distance between two atoms, and then if you push them too far, there's a very strong repulsive force. This is corresponding to electrons starting to bump into each other, essentially overlap. Those are the molecules starting to overlap, and they 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 start to push against that. If you move far away, interaction disappears. There's two molecules that are far apart, don't see each other. You can say it this way, and somewhere close to the molecules, there's a little bit of weak attractive force, and the minimum corresponds to so-called van der Waals distance between a pair of atoms. They can be decomposed into distinct radii for each atom type, and that's how those space filling models are created. Each ball corresponds to the radius of a roughly electron cloud that's limiting how close two things can get together. If you look through a slice of a molecule, this is just the same molecule as here, sliced, cut through the center. You can see that there is not much empty space there. The previous models that you've seen, the stick models in or cartoon models made out of ribbons, might make you think that proteins are empty inside. It's actually opposite. Proteins are pretty tightly packed, and there's not much space left between neutral atoms. So this excludes water from inside of the molecule, and allows water to increase its entropy, and that's what's driving the structure of the folding protein to its native conformation. So that's the fourth interaction that prevents protein from completely collapsing on itself. Atoms can't occupy the same space, but the problem is, as I mentioned, getting the accurate estimation of the energy of the protein chain is hard, and it's to a large extent because the structure of proteins is driven by not only discrete interactions between charge groups or partial charges in case of hydrogen bonds, but there's also large contribution from the solvent and modeling interaction of water with proteins is pretty hard. So there's a number of ways to approximate that, but it's essentially decreasing the accuracy of occultation of computation of the energy. This is one caveat in when trying to find what's the minimum of the free energy of a protein chain. The second problem is that finding it is hard because there's a lot of conformations to check. We've got, if we try to go blindly, let's say if we just go after the shape, the conformation of the main chain, to do that we've got to know each psi and phi angle for each residue. So you've got, let's say, 150 amino acids, then there's gonna be 149 peptide bonds, and for each we're gonna have phi and psi angles, so we're gonna end up with two psi values per bond with well, let's assume we try just 10 different values of psi angles per bond and 10 different phi values for each bond. So for one peptide bond, we've got 10 possible psis, 10 possible phis. So overall, there are 100 different combinations for one peptide bond, and now we do have about 150 of those. So then we've got 100 for the first one times 100 for a second one times 100. Overall, we do have 10 to 300 conformations to check. That's a lot. That's a lot, and this is looking just at the conformations of the main chain. If we try to even test, let's say, 1,000, let's say a million or so of those per second, it still will take 10 to 93rd. This is way beyond the age of universe or whatever. This is not feasible. The observation, the idea like that was the first observation like that was made by Sir Leventhal in a probably late 60s, I think, or so, and is referred to as a Levinthal's paradox. Clearly, proteins somehow solve the problem. They don't blindly test every possible conformation, decide what's the best one. They sample only a subset of conformations, and they can do it on its own. So one of the challenges is to find ways of stimulating how proteins fold so that you can get fast enough to a folded structure. One way to do it is to start by looking at small sections of the chain and finding out for each section what is the most likely conformation, and then use this thing for first three residues, and then look for another next three and see what's the most likely conformation there, and then you start stitching your protein from smaller segments, and then you've got the initial trial model that might be close to your to local to the global minimum, and then you try to sample conformations around that. This is roughly what Rosetta program does. That's the structure prediction program that was done that was dominant before alpha came into being. It was pretty good at predictions at a certain extent, but once more it was not solving all the problems, and besides trying to fold protein hierarchical way, it was also trying to employ information either taken from experiments or taken from looking at the statistical properties of large sets of proteins, which pairs of residues tends to be close together, which residues tend to form alpha sheets, alpha helices versus beta sheets, and so forth. So there was some sort of a combination between the energy terms originating from pure physics and pure theory and observations collected from experiment. There was in effect the energy term energy calculated by Rosetta is a mixture of physics-based terms and what's generated based on experiments. And then apart from that, there are Rosetta is trying to start from structures that are relatively close to what the prediction what the best structure, the native structure could be. It's not always perfect. So with Rosetta, you typically run tens, if not 10,000 runs, each one slightly modified, and you find you're looking for one or two best hits among 10,000 runs or so. So it's time consuming and it's not always guaranteeing to get you the right answer. But until alpha came, that was probably the best we're able to do. Now, the problem is we need predictions because we can't cope with the sheer number of sequences that are coming in. It's much easier to collect to sequence genomes than to determine structures. This is reflected by how fast the databases are growing. We do have well, now we've got more, but at the early 2003 we had about uh, we had about 200 250 million of sequences of proteins that were reported by UniProt. Okay, if you look at NR-BLAST, we had even more. Okay, those are sequences that are better characterized. NR-BLAST does have more sequences listed. Those are sequences that are observed in a metagenomes and whatnot. So there's way more. The point is there are hundreds of millions of sequences that are there, whereas when you look at the PDB, how many records you've got there, you're getting somewhere we're getting to some around, I think, 200,000 or so. The plots here are showing how many are added per year. So if you look at the recent rate, we're adding about 6,000 or so structures or unique structures for unique sequences per year, but we're adding about 10 times more of sequences. So the ratio of sequences to structures about 10 to 1 or so, and that's a problem that we can't determine structures for all the incoming sequences. So you've got to resort to predictions. On one hand, this oversupply of sequences is bad because we can't get it's too much for structure determination. But on the other hand, we're getting a large collection of sequences, each course that are related to each other. A lot of those sequences are quite similar. They are coming from species that are closely related, they didn't diverge too much, so the proteins into related species are quite similar. They might differ at few positions, but otherwise the sequence and thus the structure stays the same. So we can utilize this similarity to cope with the influx of sequences. The observation that was made quite a long time ago, probably in the 70s or 80s, where the protein sequencing and protein sequences and when and structures were starting to pick up. The observation was made that as long as you're about 20 to 30% identical in terms of sequence, the structure is gonna be of the corresponding proteins is gonna be essentially the same. The fold is gonna be the same. The tiny details determined by locally by differences in a sequence are gonna be uh, are gonna be are gonna be there, but overall structure of the protein is gonna be the same. The borderline between the identical the conservation and of structure is depending on the size of the protein. The shorter the protein, the similarities got to be higher, but typical proteins are somewhere around 100 and more or quite more residues. So this is corresponding roughly to the zone between 30 and 40%. If two proteins are about 25-30% identical in terms of sequence, their structures are most likely very similar. And this is the basis of so-called homology modeling, where what you do is you first look for proteins with known structures that are similar to your protein of interest. You end up with, if you're lucky, with collection of structures, each similar to the structure of the protein you're looking at. And then once you've got alignment between one of those proteins with known structure and your protein, you can just convert the residues in your template into residues coming from your sequence, and you're gonna have initial guess of what's the shape of your what's your shape what's your protein structure. The This conversion is relatively simple for the residues that participate in alpha helices and beta sheets mutations. There are less encountered less frequently. What's mostly changing are the loops, are the regions that connect discrete secondary structural elements. So that's harder. It's quite often done by searching through known structure for a loop with the same sequence as the one that you've got to build into your template, that has to have a sequence from the structure, from the sequence you're trying to predict the structure of. So loop modeling is more involved. And then once you've got the loops in, then you can try to actually start optimization of how individual side chains are packing within the protein core, how the loops are actually shaped. And then every time you've got the model, you try to verify it relative to experimental information you've got about your protein, and you iterate until the model you generate is agreeing with the data that you've already corrected. And that's essentially what can be done for any protein with a close homologue with the name with the determined structure. So that's common approach for the cases where there are templates. The whole approach can be automated to some extent, and there's actually a nice server that is available for homology modeling. We'll make a break now, and then we'll try to play with the Swiss model, which is the site that can be used by any of you to model a protein. If there's a homologue, you might be able to come up with a sensible model. So we'll make another break for about 3-4 minutes, and then go to Swiss model site and see how the website works. Okay, so let's stop. Okay, let's resume recording, and here's the let me get to the top version. So this is gonna be Swiss model. And this is the website that provides the access to a homology modeling software. We can start modeling. Okay, and you can either paste the sequence you're interested in, or if the sequence is in Swissprot or Uniprot, you can provide the accession. I picked up one of the identifiers from one of the sequences that one of you asked me to run the AlphaFold for. This is one of the G-coupled receptors. It's a membrane protein that's coming from a fold that's common to an entire class of receptors. And once we enter the sequence, this is the sequence here, we can start searching searching for templates. Okay, and this is gonna take a while, not that much, probably 4 or 5 minutes, but I just run a similar search for another sequence here. Here are the results at the very end of the step that you've seen starting here. We are presented with the templates of proteins with reasonable identity to the sequence you're looking at. Recently, the Swiss model added models that were already determined by AlphaFold. So for this particular protein, we do have an AlphaFold model coming from AlphaFold people with 100% sequence identity, because this is our sequence that we're interested in. So that's the template that's coming from AlphaFold. But apart from AlphaFold, we do have entire series of structures with sequences that are identical, some around 50% or so. They differ according to other scores, and they differ in terms of coverage. For the AlphaFold, that's identical sequence, we've got covered. We're covering full length of the sequence. For other templates, we're missing some of the sequence we're interested in. The structures are smaller for sequences shorter than ours. So, but it's still not bad. It looks like we're getting 88% here. If you hover over the diagram, you're gonna see that they're getting about 80 or percent of our sequence covered by the template. Ideally, would have to have the sequence covered fully by the model. Okay, so now we can pick one of those. We'll one or more. We can I'll and we can actually select something else. So if we select the second best, okay, here's our model. We can unfold the field here to get more information. We can learn where the model came from. We can see what's the sequence. We can look at different positions along the sequence, both in our target sequence and the sequence of the model, and highlight the position of the of the residue we're interested in. So this way we can inspect the template. If, for example, one is interested, we are interested in a range of residues in our sequence, and then we might check individual templates to see if any of those is covering or is more similar in this region to our sequence than others, and this would be a better starting point. It could be that in this particular case, the homology model for our region within the protein would be better than for the other models. So one can make rational choices in terms of picking up templates, and then we can pick up one or more of interest. We can you see that not all of them are different. Those two are coming from the same PDB file. They probably differ a little bit in terms of alignment, okay, or they might be different monomers in a oligomer formed by the chain. So it might be worth checking those individual files to see what's there. But in our case, we'll just check one model, and then, or well, let's pick two separate things. Okay, let's pick up two, and then build two models. And now, after picking up our starting models, the Swiss model is gonna convert the structure of the model to a structure that will have our residues of interest, our sequence mapped into the template. And now we do have our homology prediction. Okay, we do have you do have some sort of a quality score. The coloring scheme corresponds to confidence. When you hover over given position, you're gonna see in the top right corner how confident the program is in prediction. Things in red are less predicted with lower confidence. The position of the residues within this loop are less certain. This is to a large extent because this is probably the loop that differs from our template. There is a larger difference in sequence between our sequence and the sequence of the template. So that's why the program is not certain that prediction here is high quality. In other places where differences are smaller, you see less you see confidences goes higher up. The coloring scheme is similar to whatever we'll see with AlphaFold, so that's common and good. High quality corresponds to regions marked in blue. The poor quality is regions marked in red. As you see, this is yet another case of a of an online structure browser. This is a web page that shows you model, allows you to do the same things as we've been doing in Qt, but just within the browser. You can move things around. You can zoom in and out, and you can probably move things sideways. This is either left click on a mouse to rotate. If you go along the side, it doesn't work. So there's a difference between the viewer here and Chimera. In Chimera, when you were to drag on the side, it would rotate along the axis perpendicular to the screen. Here, it's not the case. So you got to cope with it by moving things in a clever way. In around other axes, you can rotate things here and then flip things back 19 degrees to have things rotated. So you've got to be more creative, but the idea is the same. You can manipulate the model on the screen. You can change the way the molecule is displayed. So here we've got the cartoon. I think, yes, that's the cartoon representation. We can make licorice. This is equivalent of sticks in a Qt, and here's a stick representation, and there's gonna be there are different coloring schemes that you can pick up and so forth. So functionalities here are very similar to Chimera. It's just a dedicated browser used to display results from Swiss model program. Once we're happy with one of the models, we can always download the files, and I can download things as a PDB. Okay, so let's get me this one. So here's my model one. I'm gonna call it, let's say where I am. Let's put it. I want to put it in my string in here. Okay, and so this is, let's call it model one. Okay, model one. Oh, that's bad. Let's call it model. No, model one. Okay, why did I have Pymol started? I don't know. Okay, here's my model one. Now let's get myself the second one. Okay, and we can download, well, we can look at it first. So here we're gonna have another model. We can, oh no, moment, where we go? Go back. We want to first look at the second one. Are we looking at the second one? Don't do it to me. Let's see. Now we're looking still at model one. To go to model two, we've got to click on the icon. So now we've got the model based on a different template. Okay, we can change the modeling to cartoon. So it's sort of similar, but who knows? So let's download it, and so in PDB format, I'm gonna move it as before to my W26 directory here. So I'm gonna move it here and call it model 2. Okay, here, let's call it model 2. Okay, and let's back off and let's try to add our AlphaFold prediction here. This is gonna be no. I don't think this model is gonna do much to the AlphaFold model. So let's get this last one going. Shouldn't take London main 3. So this is our third model. So this is what's based on the AlphaFold, and we can do the same thing. We can download in a PDB format. We can download this thing now, model 3, and I'm gonna put it in same place. Namely, I'm gonna put it in W26 Spring directory here. Okay, and then I'm gonna call it model alpha. Okay. So we've got two models based on templates picked up by Swiss model, and we've got a model based on AlphaFold prediction, which was then processed by Swiss model, but should be pretty close to whatever AlphaFold does. Actually, we can retrieve also the model coming from AlphaFold. So let's do that. To do it, we need the identifier of our protein. This is our protein. Here, either this identifier or accession from UniProt is good enough. We can now go to UniProt first. Okay, and we can paste our identifier into UniProt search page to find the corresponding record. And if we go to structure, we can go to our AlphaFold prediction. You can see it either here, or we can go to actual AlphaFold database to get the structure. We should here have in a moment the model here. Here's more information. We're gonna go over this thing tomorrow, but for a moment, I'm just gonna grab the PDB file with the model coming from AlphaFold, and then I'm gonna drop it down into our desktop and the directory for. Okay, so now we've got four structures for our sequence. There's one that's AlphaFold model as coming from the AlphaFold server. We've got two homology models that came from Swiss model based on known structures, and then we do have a model that's based on initial prediction by AlphaFold that the Swiss model side modified possibly a little bit. So let's look at those things in Chimera. Let's see how they look there, and to do that, I'm gonna now close the windows here so we're not cluttered too much, and let's go to Chimera. Here's my Chimera, and now I'm gonna open. We used to open PDB file by fetching it directly from from the RCSB or our PDB database, but we can always open a file, and then I've got to go to desktop and our directory with the models, and let's open first our AlphaFold model, and at the same time we're going to open two files. We're going to open the original AlphaFold model and the model after Swiss modeler modified it. So you've got two PDB files selected. We open them both, and here's the result. We've got two structures. We do have a cartoon representation of both. They are clearly placed in the same place, so they are pretty well aligned. If they happen to be in completely different orientations, which might happen in general, PDB orientations of the molecule, orientations of molecules in a PDB might be different. If they are not well aligned, we can always go to tools and then go to structure analysis and to matchmaker. Now we're gonna be offered with the window which allows us to specify one of the structures as a reference. This is the one that's not gonna move. So in our case, we can pick up the original AlphaFold model, or we can switch. Okay, and now we've got the other structures present in a read-into chimera that we can actually select. Here there's only one more, so we don't have much choice. We've got two molecules to align, and then using default options we can apply the alignment, and there's not much change here because our starting orientation place in a space of the two molecules was quite similar. So there's not much we can improve here, but so we'll close the window, and now the overall pos shape of the overall conformation of the main chain is similar. We still don't see the side chains. So now what we can do is we can show side chains. So to do this thing, we just flip things here, and as you can see, there's not much difference. The residues are all the residues are in a very similar orientations. Apparently Swiss model didn't make many changes. It moved things a little bit, but not much. So as you see, you can't improve much on the AlphaFold model when using Swiss modeler. You're gonna get the same, roughly the same structure. But what about the other two templates? We can read, let's say, one more. We can open and read one more molecule in. So let's get model one. Okay, and let's open this one. Okay, it's also in the same place, so we can barely see it. So let's turn off for a moment the other two, and let's oh no, well, it's somewhere here, but we don't see it well. If that's the case, it might be somewhere, but we don't see it because it's in different place uh, than the why. Oh yeah, you see, okay, here it is. You see, this one is far away, so how to align them? Well, we can go to tools, we can go to structure analysis and matchmaker. Okay, now we're gonna keep once more the original AlphaFold model as a reference, and we're gonna move the other two models to overlap as best as well as possible with the reference. And we now apply, okay, and close the window, and now we've got three models placed in the optimal position. So to now we can to declutter this thing, turn off either alpha or Swiss modular version of the model because they look the same. So let's hide the model from Swiss modeler, and now we've got in salmon the AlphaFold, in pink we've got the Swiss model version based on one of the templates. And now we start to see differences. Right now, if we turn off for a moment side chains, there are differences in a position of the helices. Apparently, they are slightly different. You do see additional sequence here that's present in the AlphaFold model, but not in a model based on homology. This is clearly because we didn't have this piece of sequence in a homology model. So in a template, this is the template that didn't have full coverage of our sequence. So we're missing those regions at the ends from the prediction. The prediction ends we can check by hovering over the chain. Prediction ends at position 40. That's the first residue modeled by Swiss model. The leading 40 residues are missing. This is probably quite reasonable. If you remember a little bit of cell biology, membrane proteins are inserted into membrane core trace of a signal peptide that's on N-terminal end of a protein. So it's quite likely that this part here up to wherever you've got the start of the homology model corresponds to a signal peptide that is used for inserting the protein into this particular protein into the membrane, and then it's clipped off. So in a final version of the protein, and this part is never present. It's clipped off during as a post-translational modification. So alpha doesn't know about biology, so alpha will try to model it anyway. So it's quite sensible to actually remove this part of the alpha model from the prediction. To do this thing, well, we can do it. That's right. We just got to figure out how to specify the region to be removed. Okay, if I hover over one of the atoms, I should be I should be able to see what this thing is. The last part, the #1/A THR17 specifies which residue I'm hovering over. This is model 1, chain A, and that's thrown 17. So to address it in a command line, to specify in a command line, I can say, let's try to color the first residue here. I can color number one, this was the number of the model. Let's see here. I hover here. Come on. This is number one, chain A. So then I'm gonna do number one slash A slash A slash A, and let's say residue one, and I want to color it red. Okay, then red. Let's see. Color, color. The other way around. Color red position. Oh, sorry. No, I'm bad. My bad. It's supposed to be color. I keep forgetting about column color. Hash one slash a column one. I think that's how it should go. Let's see. No, one, not space. No color. Oh, and I'm missing the color. Yeah, that's red and no space here. Okay, I'm dyslexic. Here we go. Okay, so this specifying the model, or in our case, this is gonna be one of the PDB files. We can read the number in a panel here. The AlphaFold came as number one. The other two are number two and three. Then we are specifying the chain, and then we're specifying either residue number or residue type, and then at the very end we're specifying the color we want to use. If we want to select entire range, then we can select this was one, this is number 40, so we want to remove 1 to 4 to 39. So let's first color 1 to 39, and this is what's not in our homology model. So if we're certain this is we want to remove, we can, instead of coloring things red, we can delete this fragment. Okay, so we trimmed out a part of the AlphaFold prediction that's not present in the final version of the protein. Okay, we don't know anything about C-terminal parts, so it might have been processed post-translationally. In case of the real protein, it might be not there in a model because it was removed. It was required further investigation to see what to do with the whisker here, but we've got a little bit better version of our AlphaFold. So now we removed part of the protein. We can try to align those things maybe better, because now our reference structure is closer to other two models. So I'm gonna go once more to tools. I'm gonna go to structure analysis, matchmaker. I'll select this one as a reference. We've got two more to align, and then we apply. They might move a little bit, they might stay the same, but just in case something could be better, and we can always rerun the matchmaker. And then here's the other model, and let's turn off the side chains for a moment so that we're not having. And the other one has the same issue. It has the signal peptide there, so this is gonna be model in blue, so it's model 2, so we can also delete number 2. Okay, so here's Here are our three modules there. I'll turn off blue out. Okay, so this is what we've got when looking at the helix at the backbone. What about the side chains? So we turn the side chains on. Okay, and then we can check what's happening along each of the helices. Okay, let's see. So let's look at these uh, two two helices here. Okay, we get more space. Let me make this thing bigger. Okay, so we do see that there's some shift in a position of a particular residue. There's a difference between where they're in a one model ended up relative to the other one, but this is the same residue. This is position 47 in a homology model versus this is position also 47 in AlphaFold. So same thing holds for all the residues along the helix. So apparently the alignment that underlied the modeling was correct. The register of the helix is correct. We don't have a position swap shifted by one or two. It seems like this is correct. What about another helix? Here's another case, and it's the same situation, right? We do have, let's say, cysteine here. This is cysteine 289, and here's another cysteine 289 from the other module. So once more, this helix seems to be also consistent between two models. So in principle, when checking models, you should check features like that. If the sequence identity was lower, there's a chance that helices were shifted by a few positions, typically by one turn. It's as I mentioned before, helices tend to have opposing faces with different properties. They're amphipathic, and then shifting by one or two positions in alignment is not likely. But if we shift by entire turn and move this histidine Y one turn away, then in some cases you might get models that are mismatching the position of the helix. So it's worth checking all the secondary structural elements for discrepancies if you're comparing between different models. And then for secondary structural elements, you'd expect as small differences as possible, but there might be differences in the loops, and I mean, you see those things here, that this loop is modeled in different way by AlphaFold. This is AlphaFold prediction. This is our homology model coming from Swiss model. So you see there are differences there, and those things might matter if you're trying to interpret your experiments based on a predicted structure. You would look for a model that as close initially to your sequence as possible. So there's it's less likely to have a loop mismodeled, and you might also try different models. So let's try another one. we do have one more in here. So let's get another file. That's the model 2. I don't think I have it yet. So let's open it. Okay. Where did it end up? Well, it's once more ended up in the wrong place. So we've got to overlay it with the other three. So that's once more tools, structure analysis, matchmaker. We once more are using the initial AlphaFold as the reference and move everything else in place. We apply, and now the green thing pops in where it should go. Uh, let's get rid of the window, move it back here, and then we've got to once more. Okay, so now let's look just at two different homology models. Let's turn off the alpha for green one, and let's turn off the sidechains for a moment. So here you see the difference in a in a in a in a in a modeling of the loop, right? In a pink model, Swiss modeler was trying to build in a piece of an alpha helix here. There is a break and then piece of alpha helix. The green model is corresponding to some other structure with no alpha helix built in here. So clearly there are differences, and then this emphasizes the importance of trying to find a model that's as close to your sequence as possible in terms of sequence similarity, especially in the regions that you're interested in, because then some of the models might be better explaining your experiments than others, and then if they explain your experiments better, then you can claim that this particular model is supported by your data. So that's one of the reasons to check more than one template, and then for looking at difference between those. Of course, I mean, you've got to check initially if there's no gross errors like misalignment of helices and things like that. And if we look now and side chains, we can show side chains. We notice that they look different, and we can see, for example, okay, so let's look here. So if we were to look at the loop here versus the loop here as an alternative, now this is presumably the same phenylalanine. This is phenylalanine 235 in model number 4, and that's 235 in model number 3. If I were to pick which one looks better, I would probably pick out green as better, because in a green model, if I turn off pink for a moment, in the green model, we do have this phenylalanine packing against another helix and possibly getting sheltered from the solvent more. In a purple version of the model, this phenylalanine sticks all the way out. Okay, and that's probably unlikely. We are dealing with the membrane protein, but this is a region probably outside of the membrane. if you look here, the this is probably what's on the outside of the membrane. This is not in a hydrophobic part of the of the membrane. So hydrophobic residue like phenylalanine is unlikely to be exposed on the surface of the protein. In the other model, if we look in the green model, it goes farther in, so it's probably more likely. The problem with this loop, though, is that this particular part seems to be completely disordered, which doesn't look right. We'd expect to have all the loops packed in some sensible way, but it could be that this particular region is participating in some sort of a protein-protein interactions or interactions of some sort with ligand. This is, after all, a receptor that binds ligands and interacts with other molecules. So it could be that once this loop is presented to a ligand, it will rearrange itself and pack on the ligand. So those are things to notice in a protein structures, and it's informative to play with the models compared. It's informative to compare different models generated for the same protein, because then you can pick up regions that are modeled consistently in the same way. You're gonna pick up models that are regions that are modeled in different ways, and those observations are informative. They can tell you something about what's going on, and you might be able to sort out things that are mispredictions. Probably what you've seen at the very beginning, the attempt to model the signal sequence within the protein structures was probably not correct, but then the difference in the loops like here might be actually corresponding to different possibly different conformations of the same part of region corresponding to ligand binding and ligand either present or absent. So that's something to pay attention to. So that's it for today. We'll continue on tomorrow with actual AlphaFold and generating predictions there. I did get this particular sequence or set of sequences from one of you together with likely ligand. So we'll see how alpha copes with the same sequence, same protein in presence of a ligand, and how what's gonna come out of there. So that's gonna be for tomorrow with some basic information about what AlphaFold actually does. It's very different from modeler, very different from Rosetta. Alpha doesn't deal with energy of a chain. It doesn't evaluate different energy terms and doesn't try to minimize the overall energy of the protein chain. The approach is completely different, and you'll see the difference tomorrow. So that's it for today. Oh, there's a chat here. Well, no, it means the question in the chat is, should I use only one database? Well, you should use as many as feasible, because it could be that one of the models that you use from is better than the others. Personally, if I had a choice between alpha and the close experimental model, I would probably get a model of based on a closely related sequence. It would be most likely a little bit better than whatever alpha is saying, but it's worth comparing. It's always useful to compare different compare different models and see which parts are consistently predicted the same way, which parts different, and then if there's if the same region shows up over and over again in a model in the same way, it's probably predicted correctly. Things that differ might be worth further scrutiny, because it might be that it's either misprediction, or it could be actually something that tells you about biology. As long as quality of the alignments, one way to check is to do whatever I demonstrated here, for checking if the secondary structure elements are aligned in a proper way. The gross problems with alignments will manifest themselves as situation where you do have in one model. Let's say if we turn two of those on. So if you've got I need a good example. Yeah, so for example here, so if you've got the two models, here we've got the same histidine located in the same, roughly in the same position, and then you've got another case. Here's an arginine starting in the same place. It's got different direction where the side chain points, but you've got this residue that's in the same place. If this charged residue ended up on opposite side of the helix, pointing towards the protein interior, it's probably a sign that the alignment is bad, that the helix got shifted by a couple of positions. So those things can happen. So it's worth to see if if residues that are charged in both sequences end up, if a charged residues end up in a sensible place, if hydrophobic residues end up in a proper place. In general, charged residues should end up on the outside of the protein, at least for soluble proteins. With membrane proteins like here, you're gonna have a region on the outside that's hydrophobic. So there's gonna be a region somewhere here that even on outside the protein is hydrophobic. But in general, charged residues should be exposed to the solvent, to the water. Uh, the residues that are hydrophobic should point inside. You might find pairs of charges inside the protein, but they should be of opposing charge. If you do see isolated positive charge inside the protein interior, it's something suspicious. Positive charges tend to end up on the surface. Unless they're accompanied by oppositely charged residue, then the charge cancels out. You've got a charge interaction. You've got the electrostatic interaction between those two residues, pulling them together. So that's sensible. But isolated charges inside the protein interior are suspicious, and those things might be manifestations of just simple errors with alignment. So that's something to pay attention to, and it's relatively straightforward to look at when you do have a model, and you look at this thing at Chimera and start looking at individual details. It's tedious, but it's the way to go. It's to compare your model with, to look at your model and see if it looks sensible. So that's about it. We'll go with AlphaFold predictions tomorrow. So I'm gonna turn off the recording. Oh no, I want to. Oh, let's see what I want to do. I want to stop recording. Yeah.